Assalamu alaikum. This is Hira Rahim. Today, the topic of our discussion is post-transcriptional modifications and difference between prokaryotic transcription and eukaryotic transcription. First of all, we will discuss the post-transcriptional modifications, and before this, we will discuss that why there is a need of post-transcriptional modifications. In case of eukaryotic transcription, after messenger RNA formation, it has to move from nucleus to the cytoplasm for further processing, for translation, for protein synthesis. So it has to move from nucleus to cytoplasm. As we know that cytoplasm contains phosphatases and nucleases, which leads to the degradation and destruction of the messenger RNA if it is not gone through the post-transcriptional modifications. So we have to, in order to protect the messenger RNA from nucleases and phosphatases, it has to protect it by transforming itself by post-transcriptional modifications. Here are the three processes which are involved in the modifications of messenger RNA. First one is capping, second one is tailing, and third one is splicing. First of all, capping. Messenger RNA adds a 7-methyl GTP cap at its 5 end. It forms a 5 to 5 carbon bonding. As 5 to 5 carbon bonding is not degraded by any of the nucleases, the 5-3 carbon bonding is prone to the nucleases, can be degraded by the nucleases, but 5 to 5 carbon bonding is not prone to the nucleases and it is not degraded by the nucleus. That's why messenger RNA form a 5 to 5 bonding with form a 5 to 5 bonding, and in order to protect it, in order to prevent itself from nucleases. Second one is 3 end tailing. At 3 end of the messenger RNA, 250 to 300 adenine bases attached, and these bases also leads to the prevention of messenger RNA from nucleases and phosphatases. Then comes the splicing, third one. In case of splicing, what is splicing? The messenger RNA contains exons as well as introns. Exons are the coding regions. Introns are the non-coding regions. So, splicing is basically the removal of introns and ligation of exons. In this case, messenger RNA remove introns and the exons are ligated, and in this form messenger RNA is formed. Exons are only the coding parts of messenger RNA, while introns are the non-coding regions. They are not required for the further processing or further formation of proteins from messenger RNA. Then we will discuss about difference between prokaryotic and eukaryotic transcription. Prokaryotic, in case of prokaryotic transcription and translation both occurs in the same compartment, both occurs in the nucleus. So they don't have to move from nucleus to cytoplasm. That's why they don't need the post-transcriptional modifications. And in case of eukaryotic transcription occurs in the nucleus and translation occurs in the cytoplasm. In case of transcription, messenger RNA is formed, and after the formation of messenger RNA, it has to move from nucleus to the cytoplasm. Second one, transcription and translation occur simultaneously. Messenger RNA formation as well as protein formation occurring simultaneously. Both processes occur side by side. But in case of eukaryotic transcription occurs first. Protein formation occurs after the completion of transcription. Messenger RNA will be formed first and then protein formation occur after the completion of transcription, because there is a stage between transcription and translation. The stage is the post-transcriptional modifications. Then comes only one type of RNA polymerase is used to transcribe all three types of RNAs. As we know, there are three types of RNAs: ribosomal RNA, transfer RNA, and messenger RNA. Okay. the only one type of RNA polymerase is required to transcribe, to produce, to code all three types of RNA in case of prokaryotic transcription. But in case of eukaryotic transcription, there are different kind of RNA polymerases which are used to transcribe the three RNAs. First one is RNA polymerase 1, which is required to transcribe ribosomal RNA, which are the subunits of ribosomes. RNA polymerase 2 is required to transcribe messenger RNA, which is used to transfer the information from nucleus to cytoplasm. They get attached to the ribosomes, and further they expose their codons, and transfer RNA finally comes and add the amino acids to form the polypeptide chain. RNA polymerase 3 leads to the formation of transfer RNA. In case of prokaryotic transcription, bacterial genes are polycistronic. These genes, in this case, one gene is capable of coding for more than one proteins. One gene can produce more than one protein, but in case of eukaryotic transcription, the genes are monocistronic. Monocistronic means one gene is capable of coding only one type of protein. Then messenger RNA doesn't require post-transcriptional modifications. Why? Because transcription and translation occurs in the same compartment in the cytoplasm. But in case of eukaryotic transcription, messenger RNA has to move from nucleus to the cytoplasm, and in order to prevent itself from degradation, from destruction, it adds cap at the 5 end, it adds tail at the 3 end, as well as it has gone through the process of splicing. Then promoter, these are the binding sites where the RNA polymerase got attached. In case of prokaryotic transcription, two binding sites TTGACA, it is present at -35 sequence. -35 means it means downstream sequence. It means there are 35 nucleotides back from the structural gene. TATAAT -10 means the sequence, this promoter sequence is present behind the structural gene after 10 nucleotides. In this case, this sequence is present -75 nucleotides. It means we have to move from structural gene 75 nucleotides, and where the promoter sequence will be present TATAAT -25. It means this sequence is 25 nucleotime downstream to the structural gene. This was all from my side. You can tell me that on what topic I have to record the next video. Allah Hafiz.